Welcome to the deep dive. Today we're getting into something huge, something often called the ultimate application of genetic technology. We're talking about gene therapy. We're drawing mainly from essentials of genetics for this one. And our mission, well, it's to really unpack gene therapy. What is it? How does it work? We'll look at its history, the highs, the lows, and definitely where it's heading, including those tricky ethical questions. It's quite a journey, from the lab bench right to the patient's bedside.
Exactly. And it's fascinating because it's not just the what, but the why. Why is it so revolutionary, but also so complex? We're basically trying to fix diseases at their source code, their genetic root. That's a massive shift from just treating symptoms.
Right, a total paradigm shift. So let's unpack that fundamental idea. What is gene therapy at its core?
Well, simply put, it's the delivery of therapeutic genes into a patient's cells.
Mhm.
The goal is to correct genetic conditions caused by, a faulty gene or genes.
So not just managing symptoms, but aiming for a cure by either adding a working gene to override the faulty one, or these newer methods, they can actually remove, correct, or even silence the bad gene.
Precisely. It's about fixing the underlying genetic blueprint. And this really embodies what we call translational medicine, taking that lab discovery, like finding a disease gene, and turning it into a real therapy. It's a cornerstone of precision medicine, too, tailoring treatment to the individual's genetics.
Okay, so who's a candidate for this? What conditions are we talking about?
Well, there are two key things needed. First, you absolutely have to know the specific gene or genes causing the problem.
Can't fix it if you don't know what's broken. Makes sense.
Exactly. And second, you need to be able to clone or synthesize that gene in the lab. You need the tool to work with.
And I guess the Human Genome Project really opened the doors here.
Oh, massively. It gave us a much clearer map of disease genes, so the number of potential candidates for gene therapy just exploded.
Initially, the focus was mostly on single gene disorders, right? Like cystic fibrosis.
Yes, those were the logical starting points, similar targets. But another big factor is accessibility. Can you actually get to the cells you need to treat?
Ah, right. So blood disorders like leukemia or hemophilia might be easier because you can take blood cells out, treat them, and put them back. Much harder for, say, brain cells.
Exactly. That's a major challenge. But despite that, the range of conditions being targeted now is, well, pretty staggering.
Like what?
Oh, everything from inherited blindness, hearing loss, neurodegenerative diseases, think Alzheimer's, Parkinson's, ALS cardiovascular issues, muscular dystrophy, HIV. Even things like depression and addiction are being explored. Cancer actually makes up about 65% of trials. There are over 2,300 trials globally.
Wow. And in the U.S., all these trials need strict approval first, from review boards and the FDA.
Absolutely critical. Given what we're doing altering genetics, the oversight has to be incredibly stringent.
Okay, so how does the delivery actually happen? How do you get the therapeutic gene in there?
There are two main strategies. The first is called ex vivo gene therapy.
Ex vivo, meaning outside the living.
Correct. You take cells out of the patient, maybe blood cells or stem cells, treat them in the lab to add the correct gene or fix the faulty one, and then transplant those modified cells back into the patient.
And the advantage there is no immune rejection because they're the patient's own cells.
Exactly. The other approach is in vivo gene therapy inside the living. Here, the therapeutic DNA is delivered directly into the patient's body, aiming for the specific affected tissues.
Okay, but that sounds trickier. How do you make sure it only goes where it's needed?
That's the major challenge with in vivo, yes, restricting delivery. But for both methods, the absolute key is the delivery system itself. DNA is big, it's charged, cells don't just naturally soak it up.
So this is where viruses come in.
Right. We use viral vectors. These are viruses that have been engineered. We take out their harmful disease-causing parts and their ability to replicate uncontrollably, and instead we load them up with the therapeutic gene. They're basically like microscopic delivery trucks that are really good at getting into cells.
Okay, and there are different types of these viral trucks.
Yes, several main types. Retroviruses, like modified HIV, are interesting. They have an RNA genome, but once inside the cell, they convert it to DNA, which then gets integrated, actually woven into the host cell's chromosomes.
So that leads to long-term expression because it becomes part of the cell's permanent library.
Potentially, yes, though that integration has its own risks, as we'll see. Then there are adenoviruses. They can carry larger genes, which is good, but many people have immunity to them already, which can cause issues.
What else?
A very popular one now is adeno-associated virus, or AAV. It's generally non-pathogenic, doesn't usually cause disease, and triggers a low immune response. Crucially, it usually doesn't integrate into the chromosome. It tends to form these little independent circles of DNA called episomes in the nucleus.
Okay, so less risk of disrupting existing genes, but maybe not as permanent, especially in cells that divide a lot.
That's the trade-off. It's great for non-dividing cells, like in the retina or brain. For dividing cells, the effect might dilute over time, needing repeat treatments. And finally, lentiviruses, which are a type of retrovirus, like HIV, they can carry fairly large genes and, importantly, can infect non-dividing cells too. They still integrate, but seem to have a somewhat lower risk of causing harmful mutations than older retroviruses.
It's amazing how they've repurposed these viruses. Are there other delivery methods being explored?
Definitely. Stem cells are increasingly important, especially hematopoietic stem cells, or HSCs, from bone marrow. You can take them from the patient, modify them ex vivo, and put them back. They're accessible, they replicate, they're long-lived, and they generate all the different blood cells. They're like programmable cell factories.
There was that incredible story about the boy with the severe skin condition, junctional epidermolysis bullosa. They used stem cells and the retrovirus for that, right?
That's a landmark case, yes. Took a skin biopsy, used a retrovirus to deliver the correct LAMB3 gene into skin stem cells, grew sheets of genetically corrected skin in the lab, and transplanted them back. Regenerated about 80% of his skin. Just astounding.
Truly mind-blowing. Are there non-viral methods too?
Yes, people are working on things like chemically assisted transfer, nanoparticles, fatty bubbles called liposomes, even ideas like gene pills. But getting naked DNA into cells efficiently and safely without it getting degraded, that remains a big hurdle for non-viral methods.
Okay, so we have the tools, but the history wasn't exactly smooth sailing, was it?
Not at all. The early days, around 1990, started with huge optimism. The first trial, widely considered a success, was for Ashanti de Silva. She had 8A-SCID, basically no immune system.
Right, the bubble boy disease.
Exactly. They took her T cells, used a retrovirus to insert the working ADA gene, grew them up, and infused them back. She did improve, though she also needed other treatments, so it was a bit complex to parse the exact effect initially.
But later treatments for ADSCID using stem cells were more definitive.
Much more so. Over 100 children have been successfully treated for a DA-SCID using HSC-based gene therapy. It's still seen as, probably the most successful example overall.
But then things took a darker turn, the late 90s.
Yes. A major setback occurred in 1999 with Jesse Delsinger. He had a liver disorder, OTC deficiency, but it was somewhat managed. He volunteered for a safety trial using an adenovirus vector.
Now what happened?
He received a very high dose, had a massive immune reaction, multi-organ failure, and tragically died 4 days later. It shook the field to its core.
Were there issues found later?
Yes, investigations revealed problems, undisclosed adverse reactions in earlier patients, financial conflicts of interest for some researchers, animal study side effects that maybe weren't fully communicated. It led to a major crackdown and rethinking of safety protocols.
Just devastating. And that wasn't the only tragedy.
Unfortunately, no. Soon after, in 2000, came news from a French trial for XSCID, another immune deficiency. Initially, it looked incredibly successful, kids developing functional immune systems. But then, several years later, 5 of the 20 treated children developed leukemia. 1 died. It turned out the retroviral vector had inserted itself right next to a gene called LMO2, switching it on inappropriately and causing uncontrolled growth of white blood cells.
Oh no, so the insertion itself caused cancer?
Precisely. This phenomenon, insertional mutagenesis, became a huge concern. The FDA halted dozens of trials using similar retroviruses. It was another massive blow.
So these terrible events really highlighted the dangers.
They absolutely did. They laid bare the key problems with the vectors at the time. 1, random integration, often only in dividing cells. 2,
Great.
severe immune reactions to large viral doses. 3, the risk of insertional mutagenesis activating cancer genes or disabling essential ones. 4, limited carrying capacity for larger genes. And 5, the potential, however small, for recombination to create an infectious virus. Big challenges.
It sounds like the field could have just died then.
It was a very difficult period, but testament to the researchers and the potential, it didn't die. Instead, there's been this incredible fast-paced resurgence in recent years.
Fueled by what? Better vectors?
Yep.
New approaches?
Both. And a much deeper understanding of the risks and how to mitigate them. We've seen some fantastic successes lately. For example, treating Leber congenital amaurosis, LCA, a type of inherited blindness.
How does that work?
They use an AAV vector, remember, the one that usually doesn't integrate to deliver the RPE65 gene directly under the retina. It helps process vitamin A. The results, especially in children who haven't lost as many retinal cells yet, have been remarkable. Many gained enough vision to navigate independently. That therapy, Luxturna, got FDA approval in late 2017, the first for a single gene disease.
That's amazing progress. What else?
Hemophilia B, using an AAV vector delivered in vivo to the liver to provide the factor IX gene. In one key trial, a single dose allowed several patients to stop needing their regular, expensive factor IX infusions. A cure feels very close for hemophilia B.
And you mentioned using HIV itself as a vector?
Yes, a highly modified disabled lentivirus derived from HIV was used successfully to treat B thalassemia, delivered the correct B globin gene into stem cells ex vivo. The patient became transfusion independent. There was some activation of another gene, but thankfully no leukemia like in the ex-SCID trial. It showed these vectors could be made safer.
And there were successes in Milan, too.
Right, for SIMLD, a devastating neurodegenerative disease, and was another immune disorder. Again, using lentiviral vectors with HSCs ex vivo, they managed to halt MLD progression and treat WAS without causing cancer. These successes were built on, 15 plus years of painstaking research.
So alongside refining these delivery methods, something else came along that really shook things up: genome editing.
Exactly. This is a different philosophy. Instead of just adding a gene to compensate, genome editing aims to go in and precisely fix the broken gene, remove it, correct it, replace it.
Like using molecular scissors.
Kind of, yes. The early tools were things like zinc finger nucleases, ZFNs, and talons. These are engineered proteins designed to bind to a specific DNA sequence and make a cut.
And ZFNs were used in HIV trials, inspired by that patient, Timothy Brown, who was cured via a stem cell transplant.
Correct. Timothy Brown received stem cells from a donor with a natural mutation in the CCR5 gene, which HIV uses to enter T cells. He was functionally cured. So researchers used ZFNs to try and deliberately disrupt the CCR5 gene in HIV patients' own T cells ex vivo, making them resistant to the virus. Showed some promising results.
And talons were similar, used for skin or muscle disorders in lab cells.
Yes, similar idea targeted DNA cutting. Both ZFNs and talons were big steps, but designing and building those custom proteins for each target gene was, well, complex and time-consuming.
But then came CRISPR-Cas. Everyone talks about CRISPR. Why was it such a game changer?
CRISPR, it really did revolutionize things. It's based on a bacterial immune system. The key is it uses a guide RNA molecule, sgRNA, to find the target DNA sequence. It's like a programmable GPS. This guide RNA leads an enzyme, usually Cas9, to the exact spot.
So the RNA does the aiming, not a complex protein.
Exactly. The Cas9 enzyme then makes a double-stranded cut in the DNA, but only if a short marker sequence called a PAM is nearby. Then the cell's natural repair mechanisms kick in. If you provide a correct DNA template alongside CRISPR-Cas, the cell can use that template to repair the break, effectively editing the gene.
And that's easier to design.
Much easier. You just need to design the guide RNA sequence to match your target. That's far simpler than engineering a whole new protein like with ZFNs or talons. It made precise genome editing vastly more accessible and efficient.
Okay, so what are some of the things CRISPR is doing?
Incredible stuff. Curing genetic liver disease in mice with a single treatment, engineering T cells for immunotherapy to make them better cancer killers.
Like Kymriah, that leukemia treatment. Does that use CRISPR?
Kymriah uses CART technology, which is related engineering T cells but often uses older vector methods. However, CRISPR is now being integrated into next-generation CART therapies and other immunotherapies to make them even more potent and specific. Trials are planned or underway using CRISPR to edit T cell genes like TCR and PD-1 to better fight tumors.
Wow. What else? Fixing Duchenne muscular dystrophy in mice.
Yes, by editing out the faulty part, the exon of the dystrophin gene, showing promise for sickle cell disease and B thalassemia by editing stem cells, curing hemophilia B in mice, disabling that CCR5 gene to block HIV, developing treatments for specific types of blindness and hearing loss. The list is growing incredibly fast.
Are there still concerns, though? Like cutting in the wrong place?
Definitely. Off-target mutations are a major focus of research, ensuring CRISPR only cuts where intended. And sometimes the repair process itself can cause unwanted dilutions or rearrangements. So safety and precision are still paramount, but the progress is just remarkable.
Okay, so we've talked about adding genes and editing DNA. What about working with RNA?
Right, RNA-based therapeutics. This is another angle, often focused on gene silencing, turning down the expression of a problematic gene rather than changing the DNA
How
itself.
does that work?
One way is using antisense oligonucleotides. These are short strands of synthetic DNA or RNA designed to bind to a specific messenger, RNA, mRNA molecule, the instructions copied from a gene and block it from being translated into protein.
And the other main RNA approach.
Is RNA interference, or RNAi. This uses the cell's own natural machinery. You introduce a small double-stranded RNA that matches your target mRNA. An enzyme called dicer chops it up into small interfering RNAs, siRNAs. These siRNAs then join a complex called RISC, find the matching mRNA, and either block its translation or trigger its destruction.
But getting these RNA molecules delivered effectively is tricky too.
Yes, RNA is notoriously unstable in the body, and getting it into cells is a challenge, similar to non-viral DNA delivery, but there have been breakthroughs.
Like Onpattro, the first approved siRNA therapy.
Exactly. Approved in 2018 for a nerve disease called polyneuropathy, it targets the TTR gene's mRNA. And perhaps even more dramatically, Spinraza, approved in 2016 for spinal muscular atrophy, SMA.
SMA, that terrible disease affecting infants.
Yes. Spinraza is an antisense oligonucleotide. It doesn't silence a gene, but cleverly tweaks the splicing of the SMN2 gene's mRNA. Splicing is how cells cut out non-coding bits and stitch coding bits together. Spinraza makes this process more efficient for SMN2, resulting in more functional SMN protein, which SMA patients lack.
And the trials were stopped early because it worked so well.
They were. The results were so positive it became unethical not to offer it to the placebo group. It's made a massive difference for many families. And this approach holds promise for other diseases like Huntington's and ALS too.
It's just incredible how many different strategies are being pursued. Looking ahead, though, what are the big remaining challenges?
Oh, plenty. Technically, we still need better ways to deliver genes specifically to tricky places like the brain or muscles. We need to figure out how many cells actually need correcting for a therapeutic effect. How do we ensure the effect lasts long term?
And what about diseases caused by multiple genes, not just one?
That's a huge leap in complexity. And controlling when and how much a therapeutic gene is expressed. Is genome editing ultimately going to be safer and better than just adding genes? Lots of questions still.
And then there are the ethical debates. They seem pretty intense.
Very much so. The key distinction is between somatic and germline therapy. Somatic therapy targets regular body cells, it affects only the individual treated, and is generally considered ethically acceptable with informed consent. That's what's proved now.
But germline therapy?
That targets sperm, eggs, or embryos. Any changes made there would be passed down to all future generations of that person's descendants. They haven't consented. That's the major ethical hurdle.
And CRISPR has made this debate even more urgent because it makes editing embryos seem more feasible.
It has. Those early CRISPR embryo studies in China, while not intended for implantation, really brought this to the forefront. But importantly, those studies also showed aging problems, low efficiency, lots of unintended mutations.
So the science itself showed it wasn't ready for prime time.
Pretty much. The researchers concluded it wasn't safe or reliable enough for clinical use in embryos. Major scientific bodies and regulators agree. In the U.S., Congress has specifically banned the FDA from even reviewing applications for germline modification.
Another huge issue seems to be costs. These therapies are incredibly expensive.
Astronomical sometimes. Glabera, that first approved gene therapy in Europe, cost over $1 million and ultimately failed commercially, partly because of that price. Kymriah, the cancer immunotherapy, is around $475,000. It raises massive questions about access and affordability. Who gets these treatments? How do healthcare systems pay?
That ties into another ethical point you raised earlier, who should be treated, thinking back to Jesse Gelsinger.
Right. His condition was somewhat managed. Was the risk of an experimental therapy justified in his specific case, even though he volunteered for a safety study? It highlights the complex risk-benefit calculations involved, especially in early trials.
And finally, this idea of gene doping. Using this tech not for disease, but for enhancement.
Yeah, that's definitely on the horizon. Using gene therapy to boost muscle growth with IGF-1 or endurance with EPO. WADA, the World Anti-Doping Agency, has banned it, but detecting it would be incredibly difficult if the gene product is identical to the natural one.
So where do we draw the line between therapy and enhancement? That's a big one to think about.
It really is.
Wow, what a journey. We've gone from the initial dream through those really tough setbacks to this current era of resurgence with tools like CRISPR and RNA therapy showing such promise. It really feels like medicine is on the cusp of something revolutionary.
It does. And history shows us this is often how medical breakthroughs happen. Antibiotics, transplants, they all had difficult early phases. There are setbacks, ethical debates, technical hurdles. But the drive to understand and treat disease pushes things forward, step by painstaking step, towards safer, more effective therapies. It's a long road, but the potential impact is just immense.
Absolutely. Well, thank you for joining us on this deep dive into the incredibly complex and fascinating world of gene therapy. We hope you feel better informed, maybe a little awed, and definitely curious about what comes next. Until next time, keep digging.